Cyanide is a famous deadly toxin that can have its fatal effects in just a few minutes because cyanide attacks aerobic metabolism. In this lesson, we're going to look at a less famous molecule, 2,4-dinitrophenol, which, like cyanide, attacks aerobic metabolism. But unlike cyanide, doctors once experimented with 2,4-dinitrophenol as a miracle weight loss drug. They soon found out that that was a mistake. Along the way, you'll be learning about how some of the major processes in metabolism fit together, because that's going to be necessary for us to see how 2,4-dinitrophenol messes those up. And you'll know that you've mastered these principles when you can describe what it means to uncouple the electron transport chain and oxidative phosphorylation, and when you can use molecular shape to help explain how a drug might have its effect within your body. Take a moment and read through this question stem right now. This prompt seems really hard. A lot of the normal evidence is missing. In fact, they haven't really given us very many clues at all. For example, they don't tell us what body system DNP works on or even what DNP looks like. So let's start there with the structure of dinitrophenol. Here's a question for you. In this blank box, we have space to consider the structure of the molecule 2,4-dinitrophenolate. So based on that name, 2,4-dinitrophenolate, what are some functional groups you can identify that this molecule will have? In fact, if you can, go ahead and draw the whole 2,4-dinitrophenolate molecule into the blank box provided. So this molecule is a phenol, meaning it has an aromatic ring and a hydroxyl group. The 2,4-dinitro part tells us that it's going to have nitro groups in the ortho and para positions relative to that hydroxyl group. And then the suffix dinitrophenolate tells us something very interesting as well. The hydroxyl group of this molecule is deprotonated. In fact, that leads us to our first prompt question. Go ahead and indicate in your book, what about the dinitrophenol molecule causes it to exist mostly in the deprotonated form in physiological conditions? There really are a lot of ways to answer this question, so I want to cover the main answers. First off, if you look at the structure of this deprotonated molecule, it really is capable of spreading that negative charge out across the whole thing. You've got both resonance effects and also this inductive effect that are pulling that charge across virtually every atom of the molecule. I always think about it like smearing cream cheese on a bagel. The more evenly you can smear that cream cheese, the better. Same thing goes for electrons. Another main answer comes from the listed pKa, given right down there. The Henderson-Hasselbalch equation in English says that if pH is more basic than pKa, the basic or deprotonated version is favored. Physiological pH is around 7 most places in the body, so that's more basic than the listed pKa of 4. Therefore, the basic or deprotonated version of this is going to be favored in your body. All right, next prompt question, and this one's interesting. You throw some dinitrophenol into the human body, and we just saw that the dinitrophenol drops a proton, making it charged. Normally, charged molecules would absolutely not be able to cross physiological membranes. Without a specific transporter, charged molecules might not even make it through your intestinal walls in the first place. But this DNP molecule is totally able to cross physiological membranes. The question is, why? Why would DNP's structure allow it to cross a physiological membrane even though it's going to be charged in your body? Well, here, DNP in general is very much nonpolar. It does have polarizing groups, but since they're pretty much at opposite ends of the molecule, any small dipoles mostly cancel. And here, remember that one negative charge is spread out over virtually every atom of the molecule. So between these factors, this guy is essentially an honorary, nonpolar, uncharged molecule. And those are exactly the kinds of molecules that cross membranes. The point is, whether DNP is protonated or deprotonated, it can get pretty much anywhere in your body just by drifting across any biological membranes that it encounters. So lastly, if we're thinking weight loss, we're thinking metabolism and energy production, what would you guess is a likely cellular target for dinitrophenol? When you're thinking metabolism at the cellular level, you're probably thinking mitochondria. And that's exactly right. We'll end up seeing that DNP completely messes up a ton of vital processes within the cell. So let's start by just naming all the processes that get messed with. Each of the colors on your screen, orange, blue, and green, each corresponds to a chemical mechanism that's going to be disrupted by dinitrophenol. See how many of these, or maybe even all three, that you can name. In a normal cell, our overall end goal is usually to make ATP, but each of these steps along the way kind of has its own goal as well. And knowing how each step contributes to the end production of ATP is a huge help in learning all these different steps. So first up, we've got glycolysis, which you can see actually occurs in the cytoplasm outside the mitochondrion. The goal here is to make pyruvate, which then moves into the mitochondrion, where it gets converted into acetyl-CoA. The process for breaking down fats is called beta oxidation. And so kind of the goal of either sugar or fat breakdown is acetyl-CoA production. Acetyl-CoA then feeds into the citric acid cycle, and the goal of the citric acid cycle is to output NADH and FADH2. These are high-energy electron carriers. So we're not quite ready for ATP yet, because those high-energy electron carriers feed next into the electron transport chain. And here, I'll hand it back to you. What is the immediate goal of the electron transport chain? As electrons move down the chain, it pumps protons into the intermembrane space. Think about what we've accomplished so far. We had energy stored for the long term in chemical bonds in sugars and fats. Fats are really stable. Glucose is pretty stable. But as we start breaking those molecules down, we get acetyl-CoA. That's a molecule that's moving around a lot, that's doing stuff in your body. So it's already just a little bit less likely to hang around. Then break that guy down even further and use the electrons for NADH and FADH2. Now we have energy stored as high energy electron carriers, but those electrons just keep passing the buck. They pump protons. And now energy is stored as a proton gradient. What we have here is energy. We're just changing forms. And each time we change forms, we get a little bit more unstable form of energy storage. Here's one more question for you about protons. These protons cannot cross a lipid bilayer on their own. Why not, and how do they cross this lipid bilayer? The protons may be exceptionally small, but they're charged. And unlike dinitrophenol earlier, protons can't spread that charge around or stabilize it or mitigate it in any way. So protons need some kind of transportation across the lipid bilayer. And the transportation mechanism that evolution chose is the ATP synthase protein. This protein harnesses the energy of the proton gradient, and our last energy transfer is now complete. The ATP synthase uses that energy to take ADP and free phosphate and synthesize it into ATP. This process is called oxidative phosphorylation. One more question for you here. Take a big, deep breath in. The same oxygen you just inhaled will be used by the mitochondrion here. So what is the role of oxygen in this whole process? Oxygen is the final electron acceptor. That means that without oxygen, the electron transport chain would have no place to put its used up electrons. So NAD and FADH2 build up, and pretty soon the whole process just shuts down. In fact, if you consider the overall net fully balanced reaction for aerobic metabolism, that overall reaction is actually almost exactly the same as the combustion reaction of something like gasoline. Both reactions have a carbon-based molecule and oxygen as their inputs. Both reactions have water, CO2, and energy as their outputs. Your body burns sugar and fat in a way that is at least chemically somewhat similar to how your car burns gas. So that's a great overview of metabolism. On test day, you might need to know like detailed facts, individual enzymes, inputs and outputs, the nitty-gritty of these individual mechanisms. But you can think about this diagram as like a table of contents for the primary reactions for metabolism. It's really nice to see how all these fit together. And in fact, keeping this big picture in mind is going to help you learn the nitty-gritty of these mechanisms as well. So now let's think about how dinitrophenol messes with that big picture. My question for you. We know that deprotonated dinitrophenol can cross membranes, like the outer membrane of a mitochondrion, and upon entering into the mitochondrion, dinitrophenol will become protonated. Why? This is basically Le Chatelier's principle at work. If you've got a weak base like dinitrophenol and you surround it with protons, Le Chatelier says that it will push toward the side where it's picked up a proton. It never ceases to amaze me that here we are at the edge of undergrad biochemistry, verging into drug design and real actual medicine, and we're still using fundamentals like Le Chatelier's principle. Next question, and this might take some serious thought. Why does dinitrophenol moving into the mitochondrion and picking up a proton, why does that disrupt ATP formation? Your first thought might have been, well, there will be less protons available for oxidative phosphorylation. And that's true, but the full mechanism is much more insidious. Remember, dinitrophenol can cross membranes whether protonated or not. So the protonated form will now move into the inner mitochondrial matrix. And then, because DNP prefers to be deprotonated, and because there's no more surrounding protons, DNP will release that proton. That proton has just completely circumvented ATP synthase. But wait, there's more. The deprotonated DNP can now move back into the inner membrane space, pick up another proton, and so on. Just a relatively little bit of dinitrophenol can completely disrupt the production of ATP. So the last thing to cover is why DNP is effective as a weight loss drug. Think for a second. The cell is no longer producing as much ATP. The cell needs ATP, so how will it respond to this drop in ATP production? The answer is that the cell will try to make more ATP, and it does this the way it always does, by oxidizing sugar and fat. In fact, not only will the cell continue to burn sugar and fat, it will actually upregulate sugar and fat metabolism. Sounds like an effective way to lose weight, but this comes at a great cost. The original question stem also mentioned that DNP causes tachypnea, which is elevated breathing rate, and hyperthermia, an elevated body temperature. Given everything we've covered, explain these two side effects. Keep in mind that the final electron acceptor in the electron transport chain is oxygen. Without oxygen, all of aerobic sugar and fat metabolism shuts down. So at the same time that your body signals for greater sugar and fat metabolism, your body also signals a greater demand for oxygen, which manifests as an increased rate of breath. As for the hyperthermia, the energy generated by burning fat and sugar is usually put into ATP formation. And then ATP usually uses that energy to do stuff in the cell. In this case, though, that energy is just wasted. And wasted energy takes the form of heat. At this point, if you feel comfortable enough with the processes of metabolism that you can start messing with them, that, for example, you can describe what it means to uncouple some of these important processes, then you've really got this material down. Your next step is going to be to learn the individual steps of each of these mechanisms so that you have both the big picture view and the very detailed view.